Dear students, this is the last lecture of unit first, which is having the energy rich compounds, their classification and biological significance of ATPs and cyclic AMPs. So, in this lecture we have to discuss what are the energy rich compounds, how they react and what is ATP, CAMP and other compounds here. So, what are energy rich compounds, how they can react? Energy rich compounds are basically those compounds which having the certain compounds in the biological system which yield energy on hydrolysis. These substance which possess the sufficient free energy which in the liberated at least 7 kilocalorie and at the 7 kilocalorie per mole at pH 7. So, those substance which having the sufficient free energy to liberate at least 7 kilocalorie per mole at the pH 7, that is the neutral pH. There is the liberation of energies around 7 kilocalorie per mole is showing the energy rich compound, and those compounds which are showing the liberation of energy less than 7 kilocalorie per mole at 7 pH or neutral pH are known as low energy compounds. So, those compounds having low energy less than 7 kilocalorie per mole are known as energy low compounds, low energy compounds. What are the high energy phosphates? High energy compounds. These are the different phosphates or different compounds which are having the total amount of energy of whenever they are undergo hydrolysis and yield energy here. So, these are the intermediate products which are formed during the metabolism of carbohydrates and other biomolecules. So, these compounds are generally liberate the different type of energy here. So, phosphoenol pyruvate which is also reacted in the glycolysis. So, these is the intermediate product which is whenever it is hydrolyzed, it gives the energy 14.8 minus 14.8, that is they are showing the high energy phosphate bond. And the second one is carbamoyl phosphate. Carbamoyl phosphate whenever undergoes hydrolysis process, it gives the energy minus 12.3, which is also showing the high energy phosphates. The third one is cyclic AMP, that means cyclic adenosine monophosphate, which is undergoes into the hydrolysis process, it gives the energy around 12 calorie per mole. After that, there are pyrophosphates which are showing the energy 8 calorie per mole after hydrolysis. There are acetyl coenzyme A also present in the reaction of lipid metabolism. These acetyl coenzyme A, when reacted, undergoes hydrolysis and they gives the energy having minus 7.7 calorie per mole. So, the next one is ATP. When ATP is undergoes hydrolysis to form ADP here, adenosine triphosphate to adenosine monophosphate here, and the production of one phosphate bond here. This is the formation of this energy is the minus 7.3 calorie per mole. So, these all are having high energy phosphates bond which are responsible for the giving energy, and these are known as high energy compounds, and they are mainly convert energy into the form of ATPs here, and these ATPs are required for the formation or normal functioning of body. Basically, in brain, then brain needs more energy as compared other organs present in the body. So, these are the high energy phosphate compounds which undergoes hydrolysis to form the final products or break down the different chemical bond to form the energy here. And after these high energy phosphate, there are some low energy phosphates also present here. These are different low energy compounds which whenever hydrolyzed to give the compound having the low energy. Here is when adenosine diphosphate when convert into the adenosine monophosphate, and there is production of release of one pyrophosphate bond. Here there is formation of 6.6 minus 6.6 calorie per mole here. So, which is a low energy phosphate. There is also fructose 6 phosphate, which is also intermediate product of the carbohydrate metabolism, which gives 3.8 calorie per mole energy in hydrolysis. And when the glucose 1 phosphate is undergoes hydrolysis, it gives minus 5 calorie per mole energy, and glucose 6 phosphate, which is also a intermediate product of glucose metabolism, which gives minus 3.3 calorie per mole energy on hydrolysis. So, these all are low energy phosphates. So, they are a very low energy phosphate compounds which are giving the energy whenever undergoes hydrolysis. What is the classification of these high energy compounds? These high energy compounds having different 5 categories, which are described here for the classification of these high energy compounds. So, there are various groups which are determine these high energy groups, high energy compound. The first one is pyrophosphate, the second one is acyl phosphate, and the third one is enol phosphate, fourth is thioesters, and the fifth in phosphogenes. And how they are reacted and what is the structure of these high energy compounds is discussed in the next. This is the general bond energy compounds. In this bond energy compound, whenever we started with the pyrophosphate, so in this pyrophosphate class, there is one carbon is reacted with the more than 2 or 2 phosphate bonds, so that these phosphate bond when hydrolyzed give the high energy compounds. So, these pyrophosphate class, which is having the different phosphate group in their structure, is when hydrolyzed in any reaction or metabolized in any reaction metabolic process, they are converted into the high energy compounds or release a high energy here. Then the example of this pyrophosphate is ATP or pyrophosphates. They are present, they are released in the Krebs cycle, oxidative phosphorylation process. These are the basically responsible for the releasing high energy compounds. The second one is acyl phosphatase. Acyl phosphatase has a carboxyl group here, and with this there is attachment of phosphate group here. This is the symbol of the attachment of high energy bond with that phosphate group. So, this acyl phosphatase is required for the formation of high energy compounds here, and the example of these acyl phosphatase is 1,3 biphosphoglycerate, carbamyl phosphate, and acyl phosphatase. These are the intermediate product which are synthesized in the metabolism of carbohydrates, lipids, and proteins. And these are the next class here for the here in high energy compounds is the enol phosphatase. In this enol phosphatase, there is attachment of 2 carbon atom here and ethylene, and there is a formation of this hydrogen atom with this high energy phosphate bond. This the symbol of this high energy this form is the attachment of phosphate with the any other group. So, this enol phosphate is when reacted with this, but the compound they are showing the high energy compound, or they are liberating high energy in the breakage of the bond. So, the enol phosphatase, the example of enol phosphatase are in phosphoenol pyruvate. In phosphoenol pyruvate, they are required for the formation of energy. The next class is thiol ester, which is also known as thioesters. These are basically important class because they are responsible for the metabolism of lipids, and they are also helpful for the metabolism of carbohydrate in Krebs cycle or beta oxidation of fatty acid, because the example of these are acetyl acetyl coenzyme A or acyl coenzyme A. Acyl and acetyl coenzyme A are synthesized in the fatty acid metabolism, where the beta oxidation of fatty acid occurs and the production of acetyl coenzyme at the last, and acyl coenzyme A in the different intermediate reactions here. So, thioesters or thioesters are having the presence of sulfur bond here, and these are basically responsible for the formation of high energy generation and formation of more than 7 calorie per mole energy. The next is guanidinophosphate or this is phosphagenes. This is also known as phosphagene. So, this is having the nitrogen when reacted with the high energy phosphate bond, and the example of these guanidophosphate या phosphogene class is phosphocreatinine and phosphoarginine. This is the basically responsible for used for the metabolism of amino acid, because there is presence of arginine and creatinine, which is the used for the in the urea cycle and in the metabolic process of amino acids. And what are high energy bonds? After determination, after having knowledge of the high energy compounds, high energy molecules, we have shifted towards the high energy bonds. What is high energy bonds? That bonds which possess acidic anhydride bonds, which are formed in the condensation of the acidic group of related compounds. These bonds are called as high energy bond because free energy is liberated when these bonds are hydrolyzed. These bonds are having energy in their structure, and when these bonds are hydrolyzed, then the free energy is liberated in these reactions, and the formation of high energy more than 7 calorie per mole. And these the symbol, this is also I have cleared in the starting. The symbol, this is used to represent the high energy bond. When there is carbon, nitrogen or and the other molecules are attached with the high energy phosphate bond, this symbol is used for determination of high energy bond. Like this, when ATP having attached with the phosphate bonds, this is showing the symbol of this, which is showing the high energy bond present here. So, now the main high energy compound is ATP, which is the main characteristic compound for the high energy substance, and it is the most important high energy compound in the living system. The mostly all the cell, all the organs use energy in the form of ATPs only. So whenever there is formation of GTP, formation of NADH, formation of FAD in the various metabolic reaction in the body, like in metabolic reaction of the carbohydrate, lipids, proteins, amino acid, these all NADH, FAD, GTP is now converted into the ATP, so that it can easily used by the different organs or cells in the body. One NADPH is converted into the 3 ATP, one FAD is converted into the 2 ATP, and one GTP is converted into the 1 ATP for the utilization by the cells and organs. So, this ATP, the name itself says the adenine triphosphate. So, adenine is the structure is of adenine, which is reacted with the ribose sugar and a triphosphate moiety. Then this is the structure of ATP here. Here is this is the adenine, which is reacted at the second carbon of the first carbon of this sugar moiety here, and when the sugar moiety is attached with this fifth carbon here, and there is formation of attachment of high phosphate bond. Yani there is 3 phosphate bond is attached here to form a ATP molecule. In this ATP molecule, there is presence of one adenine, there is presence of one sugar molecule, there is presence of 3 pyrophosphate bond here for the formation of this ATP molecule. what is the significance of this ATP molecule, how we can use these ATPs in the body. So, whenever there is hydrolysis of ATP is occurs in the body, there is release of large amount of energy. Whenever ATP is reacted with the water molecule or undergoes hydrolysis, it is converted into the ADP. There is the removal of one pyrophosphate bond, and there is the release of energy in the form of 7.3 calorie per mole. So, the energy liberated is used for the various processes which occurs in our body, like muscle contractions, active transport, function of the brain, and the other cell molecules of transcription, replication and other reactions in the body. Act as a donor of high energy phosphate to the low energy compound. It is make the energy rich compound, because the when ATP is converted into the low energy molecule, when ATP is converted into the ADP, it is released the energy and converted to the low energy compound. They form the energy of compounds to, so that the organ can use these energies for their normal functioning. Whenever there is ADP which is released here, whenever ATP undergoes hydrolysis to convert ATP here, when this ADP is again accept high energy pyrophosphate bond from the compounds, then it convert into the high free energy to form ATP. So, whenever ADP having the high energy phosphate bond from the compounds, from the possessing high free energy content, so to form the ATP here. This reaction can be reversed whenever there is presence of one pyrophosphate bond in the reaction. So, this reaction can be reversed and there is formation of what ATP here. So, there is no hydrolysis, there is a formation of ATP from ADP and pyrophosphate bond, pyrophosphate group to form a ATP. What is the biological significance? How we can determine the ATP for their use? So, it is ATP is the constantly utilized and regenerated, because we have already discussed in the last slide, ATP is when hydrolyzed to convert the ADP and pyrophosphate group, and when the pyrophosphate group is reacted with the ADP molecule and there is formation of ATP. So, this ATP is constantly being utilized and regenerated, and it is represented the ATP ADP cycle here, because once a ATP is utilized and after that then when is pyrophosphate bond present, pyrophosphate group attached with the ATP to form the ADP here. So, this reaction or this cycle is known as ATP ADP cycle, which is fundamental basis of energy exchange reaction in the living system. It is very required for the release of the energy. So, ATP is basically act as an energy link of energy between the catabolism, where the degradation of molecule occurs, or the and the anabolism, where the synthesis of the molecules occur in the biological system. So, ATP work as the energy link here. Here in energy link, this catabolism and anabolism reaction occurs simultaneously, and we can use energy in these reactions. Synthesis of ATPs in two ways. First is the oxidative phosphorylation and the second is substrate level phosphorylation. Oxidative phosphorylation, which occurs in the mitochondria, which is a major source of ATP in aerobic organism, and it is linked with the mitochondrial electron transport chain, which is also described as electron transport chain ETC. Substrate level phosphorylation, which is synthesized during the substrate oxidation in the metabolism. There is a high energy compounds like phosphoenol pyruvate and 1,3 biphosphoglysate and the succinyl coenzyme A, which can transfer high energy phosphate to ultimately produce ATP. This is a general reaction where the oxidative phosphorylation and substrate level phosphorylation release phosphate group here, and this phosphate group is reacted with the ATP to form ATP here, and this ATP is when release phosphate to help the to help energy to for muscle contraction, active transport processes or biosynthesis biosynthesis in the living creature, and the phosphorylation reactions. So, this is the basic synthesis process of the ATP, where this ATP and ADP bonds are reacted and formation of ATP occurs here, and this whenever there is a phosphate bond released from this ATP, their creatinine is reacted here and creatinine phosphate form, and this phosphate is released from the creatinine, and creatinine is again reacted with the reaction. So, there is a pathway or cycle for the formation of ATP here. And what is the cyclic AMP? What is the cyclic adenosine monophosphate here? It is a cyclic nucleotide which is chemically is 3,5-adenosine monophosphate. What is it? It is synthesized in the tissues from the ATP under the influence of adenyl cyclase whenever there is the presence of magnesium ions also. This cyclic AMP when decomposed into the AMP, and this catalyze the enzymes phosphodiesterase bond enzyme phosphodiesterase. This CAMP is a second messenger, and it is used for the intracellular signal transductions, and it is also used in the transferring the effect of hormones like glucagon and adrenaline, which cannot pass through the cell membrane. So, they are also helpful in the transferring the effects of these hormones like glucagon and adrenaline, which are not transferred or passed through the cell membrane. And the cyclic AMP is involved in the activation of protein kinase and also regulate the effects of adrenaline and glucagon. It is also regulate the passage of calcium ion through the ion channels. For this is also helpful in the contraction of the muscles, and CAMP and its associated kinase enzymes also functions to several biomolecule processes, which is also includes in the regulation of glycogen, sugar and lipid metabolism by the activating protein kinase. There are various functions of the cyclic AMP. This is the mediator of the hormone action, and it is also activate protein kinase A. This is this CAMP also help in the normal function of glucogenolysis, which is the whenever the glycogen is converted into the glucose in the fasting situation, and it is also helpful in formation of lipolysis or degradation of lipids here, and it is also modulate the transcription process or there is formation of protein. It is also helpful in the activate the hormone secretion like glucagon, like adrenaline also. It is also increases the gastric secretion. It is regulate the cell permeability in to water. It is also helpful for the permeability to cell permeability to water here. And this is the general structure of AMP and CAMP. This is the general structure of ATP, and when is ATP is reacted with the adenyl cyclase and pyrophosphate bond, there is a release of 2 pyrophosphate bond from this ATP, and there is formation of CAMP, which is a cyclic adenosine monophosphate. There is a formation of a cyclic bond here, and there is a formation of cyclic AMP. And what is the biological importance of CAMP? It is reduces the inflammation. It is helpful in the reducing the inflammation in the body, and it is also helpful in the controlling blood pressure of the body, and it is also having anti-glaucoma effect, showing the positive inotropic action in the heart, that means the increasing the activity of heart here, and it is prevent the platelet aggregation. It is help in the controlling the platelet aggregation, that mean it is showing the antithrombotic action, and it is used for the bronchodilation process. In they are showing the dilation of the bronchial cells. So, they are also helpful in the asthma condition or any type of lung related disease. So, these are the basically biological processes of cyclic AMPs, and in this process we have already discussed the structure of CAMP, biological importance of CAMP, and the structure of AMP here. So, in this lecture we have already discussed about the structure of AMP and their cyclic AMP, and synthesis of AMP, ATPs here. They are the biological significance of these ATPs here. So, we have already discussed in this lecture about the high energy phosphate bonds, low energy phosphate bond, ATPs, ADPs, cyclic AMPs here. So, these are this is the general summary of this lecture here. So, it is all discuss about the high energy phosphate bonds, CAMP and ATPs. So, thank you.